Alright, so in the last video clip we sort of ended with an introduction of photoinhibition, and just to remind you, if we go back here, we can see on this photosynthetic light response curve that as light intensity or photon flux density increases, photosynthesis increases up to a point until it reaches what we refer to as, we wrote over here, photosynthetic capacity. And beyond sort of a maximum light intensity here, oxidative damage can occur to various components of the light reaction machinery, leading to photoinhibition. And so we're going to talk about plants, or how plants can minimize or repair photoinhibition, the damage from photoinhibition. How they can minimize photoinhibition or repair the damage from photoinhibition. And also, looking at plants that are exposed to different light intensities on a regular basis that they've adapted to, such as shade versus high light intensity adapted plants, and how that affects these light curves. So that's kind of what we'll focus on here. So the question is, how does the plant prevent or repair the damage from photoinhibition? How do plants prevent photoinhibition or repair damage from photoinhibition? And we primarily talk about the first part of this question here. Okay, so the first mechanism here in place is referred to as the D1 repair cycle. And if you recall, D1 is, there's a D1 protein complex and D2 protein complex within photosystem 2. So this is a protein in photosystem 2. And we'll start by saying that the components of photosystem 2 are basically more vulnerable to this oxidative damage. Alright, so photosystem 2 is going to have a few more mechanisms here in place. And because photosystem 2 activity has to be balanced with photosystem 1, then these are going to impact the activity of photosystem 1. So the idea here is that the D1 protein, basically has a maximum number of photons that it can absorb. So when it has a maximum number of photons to absorb. And when it reaches that maximum, or close to that maximum, it dissociates from the reaction center of photosystem 2. So once it dissociates, then a new D1 protein associates with the reaction center of photosystem 2 and is basically replacing the old one. So this prevents oxidative damage on a regular basis. Another method or mechanism in place are that there are protein kinases within the thylakoid membrane. And these protein kinases nearby photosystem 2, they will sense the ratio of reduced to oxidized form of plastoquinone. So the PQH2 to PQ ratio in the thylakoid membrane. In that pool that we mentioned, we called it a PQH2 to PQ pool. And so if the reduced, we'll just abbreviate it here, to oxidized PQ ratio in the pool is high, meaning there's too much PQH2, then this is going to slow the rate of electron flow from PS2 to PS1. In other words, there's not enough of this oxidized form available to bind to the D1 protein and get ready to accept electrons coming through the reaction center of photosystem 2 from phosphates and from the quinone molecule. So when there's too much reduced form of PQH2, then it's interpreted as that there's excess energy, and as a result, the light harvesting, the protein kinase will bind to the light harvesting complex and phosphorylate that light harvesting complex. So it's, the light harvesting complex is phosphorylated by the protein kinase, which causes it to dissociate, the light harvesting complex to dissociate from photosystem 2. What is the effect of the light harvesting complex dissociating from photosystem 2? Well, there's a reduced or limited capacity to absorb energy in photosystem 2, which allows photosystem 1 to catch up, to balance the electrons moving through. So essentially it's going to cause the reduced to oxidized form of PQ to decrease or reach that optimal level again, decrease back to optimal. Or, in other words, increase the oxidized form so that it can then be available again to, more PQ can be available, plastoquinone in its oxidized form, for accepting electrons from photosystem 2. Now, how is it that the light harvesting complex can basically dissociate and move away? Well, the light harvesting complex, like the reaction center, has hydrophobic components that hold it in position in the membrane, but it does allow it to move laterally through the membrane. So that once the reduced to oxidized ratio of plastoquinone reaches that optimal level again, then the light harvesting complex, or a new light harvesting complex, can then reassociate with the reaction center and carry on. All right, so a third method, so we've talked quite a bit here about the D1 repair cycle and the light harvesting complex with its protein kinases that sense the ratio of reduced to oxidized form of plastoquinone. Another mechanism or process in place to prevent photoinhibition is the presence of beta-carotene. And beta-carotene is a pigment molecule in the light harvesting complex. It's an accessory pigment. It can absorb light energy and pass it on through radiationless transfer to the photosystem reaction center of chlorophyll molecules. But it also has this added feature where it can absorb excess energy to limit the risk, or to limit photoinhibition, or to limit oxidative damage that will result in photoinhibition. And so we're just pretty brief in that explanation there. Another mechanism for reducing oxidative damage or preventing oxidative damage is called non-photochemical quenching. And non-photochemical quenching is where excess energy dissipates as heat. And that was one of the 4 fates of energy transfer or excited energy in an electron that we discussed back within chapter 6. So this results in thermal deactivation. Another mechanism that we also talked about in that discussion is fluorescence, where after a certain amount of thermal deactivation, the chlorophyll A molecule can dissipate excess energy by red light emission, emitting that excess energy as a red light. And this is happening in chlorophyll. All right, another way to prevent excess light energy from causing photooxidative damage is this concept of stacking and unstacking the thylakoids. Unfortunately, I have this little WIN tag following my pen, so I'm not quite sure how to get rid of that right now, but we'll tolerate it for the rest of this recording. So we can see, you probably remember in looking at diagrams of the chloroplasts, there are stacks of thylakoids called grana, and so the stacked regions are shown in this area. They're also called the oppressed regions of the thylakoids in your textbook, where this would be the non-oppressed regions. And if you looked carefully at the non-oppressed or unstacked regions, you can see that we have all four components of the light reactions, the ATP synthase complex, the photosystem II complex, photosystem I, and also cytochrome B6F. Now if we compare that to the oppressed or the stacked regions, we can see all four of those components lining the exterior edges here, the top and bottom of the grana, but the interior folded sections lack photosystem I and ATP synthase. And so the question is, what would be the advantage of stacking to eliminate those components of the light reactions from those regions? Well, in the unstacked regions where you have all four, there's going to be an increase in photosystem I and in ATP synthase activity relative to, let's say, maybe more balanced by photosystem II and the cytochrome B6F complex. Whereas if photosystem II is not producing, is sort of running behind on its electron transport, then it's more advantageous to stack these here so that we have more photosystem II activity and less photosystem I activity, for example. And so why is it that photosystem I and ATP synthase is limited from these stacked regions? Well, if you look carefully as to where they are and what they're in contact with, these membranes here have the lumen inside but are exposed to the stroma on the outside. Here in the stacked regions, there's a lack of stroma, spanning. We have basically lumen on one side of the membrane and very tightly adhered to the next membrane of the stacking before we encounter more lumen. So these stacked regions have limited access to the stroma, and ATP synthase, with its requirement to be in contact with the stroma for producing ATP, and for photosystem I to be in contact with the stroma to make NADPH, then we're only going to find those on the edges here. So the stacking and unstacking is a dynamic process to help increase the rate of electron flow through photosystem II relative to photosystem I when photosystem II is not active enough. Now keep in mind that remember that the ATP synthesis and NADPH, of course, are going to be, here's the ATP synthesis, and NADPH is going to be transferred on to the Calvin cycle. And so the activity of the Calvin cycle may also govern the arrangement of these thylakoids. So as more ATP and NADPH are required, there may be greater unstacking as well. So a couple different reasons why we have that dynamic influence of these different aspects of the light reactions in terms of what the demand is and how their morphology changes. So we're going to continue on in the next video clip to look at some adaptive morphology as well as light versus sun adapted plants.